Okay, I hope everyone had a happy Easter weekend. A reminder that our review sessions for exam number 4 will be Friday afternoon, Saturday morning, and Saturday afternoon. That exam opens a week from today. So today we have just 2 more things to talk about in regards to nucleic acids, the sequencing of DNA. We'll briefly outline how that process is performed with an emphasis on PCR. Then we'll discuss viruses, different types of viruses, and how those viruses are able to replicate themselves. And then we'll go into our next chapter on metabolism, starting out with a discussion of oxidation and reduction reactions and how those are performed in nature, as well as discussing a couple of metabolic pathways, fatty acid beta oxidation, as well as glycolysis. So DNA sequencing is an area in which there have been rapid advances in the past few decades. And that process in general, because DNA is such a large molecule, the way DNA is sequenced is it is chopped into smaller pieces, and then those smaller pieces are sequenced. And then because there's usually some overlap, different molecules are cut in slightly different places, so there are overlapping sequences, and computer software will analyze where those sequences overlap and put the pieces together to give you the entire sequence of the, the DNA molecule that is being sequenced. So the enzymes that perform the cutting of the DNA are known as restriction endonucleases. And there are different restriction endonucleases that are specific for cutting the DNA in certain locations. Some of those restriction endonucleases will cut directly across the double-stranded DNA, giving what's known as blunt ends to the new and smaller double-stranded DNA pieces, whereas other restriction endonucleases will cut in different spots along the 2 strands, giving pieces that have what are known as sticky ends. And then once those smaller pieces have been generated, before the sequencing can be performed, the DNA has to be amplified. So typically in the samples that are used for DNA sequencing, only very small amounts of the DNA are present, right? So in, of course, COVID testing, this is one of the more common applications that we've had in the last few years. We have small amounts of the sample. Forensic science work with crime scenes, typically very small amounts of that genetic sample is present. And so the DNA must be amplified first before it can be sequenced. And that's where the polymerase chain reaction, or PCR, comes in. That is the process by which those DNA fragments are amplified. And it was this technology that has enabled all of these different applications of DNA sequencing, from determining what virus you have when you're sick to determining who committed a crime. And this was developed by a scientist named Kary Mullis, and he won the Nobel Prize in 1993 for developing PCR. So here is a figure showing you the basics of how PCR amplification works. So at the start of the process, there is a sample that has a variety of these double-stranded DNA fragments. But the cellular machinery, all of the enzymes that unwind and copy the DNA, are not present in the system. Just a DNA polymerase that's able to do the copying, but not all the unwinding and all the things that we talked about a little bit on Friday. So in this case, we rely on heat. If we can heat the sample up, we're able to use the energy from the heat to unwind the double-stranded DNA and give us single-stranded pieces. And then there are primers included in the mixture. These primers are small pieces, short pieces of DNA that are going to hydrogen bond to an end of these short segments. And that's going to prime the system for the replication process. And these primers are selected based on the restriction endonucleases that are used. Those restriction endonucleases will cut in certain locations and give you certain nucleotides at the ends of the chain. And so you select the appropriate primers based on the restriction endonucleases you're using. So those primers will bind via hydrogen bonding and pi stacking to the ends of those fragments that you have. And that primes the system for the replication process. And so then, once those primers are attached, then, do you have a question back there or just, okay, no worries. So then a DNA polymerase enzyme will catalyze the extension of that new chain onto the primer. So the solution in which this amplification is being performed must contain ample quantities of the triphosphates of each of our 4 nucleotides, right? We're going to have A, T, C, and G triphosphates in that solution so that the enzyme is able to attach those to the primer and therefore the growing chain. And so this depends on a DNA polymerase enzyme. We don't have all the enzymes involved in the replication process inside the cell doing this, but we have a polymerase which is attaching these individual nucleotides. And a very common polymerase that is used is called Taq polymerase. This one is very, very useful because it is stable to heat. This PCR process requires heating during step 1 in order to unwind the double-stranded DNA. And so we need an enzyme that is going to be stable, an enzyme that's not going to denature during that heating process. And so once the polymerase is done, what we've done is we've taken an initial double-stranded DNA segment and we've converted it into 2 double-stranded DNA segments, but typically we need to much more than double the amount of DNA present in the sample before the sequencing can proceed. So this cycle is repeated over and over and over again, meaning that the automated instruments that perform this go through various heating cycles. They will heat at certain parts, certain time frames in order to unwind the double-stranded DNA and then cool back down to a lower temperature and allow these processes to proceed and then heat again and so on and so forth until sufficient cycles have been performed and there's enough of the amplified DNA present in the solution for the sequencing analysis to proceed. Okay, question? Because of the chemistry we talked about in the replication process on Friday. We said that there's an SN2 reaction that proceeds. You have the 3' OH on your growing chain attacking the triphosphate, knocking off the diphosphate or the pyrophosphate. Like when we learned in our lipids chapter that pyrophosphate is an excellent leaving group. And so you have to have that knocked off of your triphosphate before you can build the backbone. Okay, so it's just SN2 reactions over and over again, building these new chains of the DNA. Any other questions about the polymerase chain reaction? Okay, so let's talk now about viruses. So viruses fit in this chapter because viruses contain nucleic acids. Viruses are going to be comprised of DNA or RNA plus a protein coat. So there is a coat or a shell of proteins encapsulating DNA or RNA. And then that virus can insert itself into cells, and then it opens up inside the cell and is able to use the cellular machinery in order to replicate its genetic material. So viruses are unable to reproduce or replicate on their own, They don't have all the enzymes and the machinery necessary to do so. But once they get inside of cells, they are able to make copies of both their genetic material and their proteins, which then allows viral particles to assemble inside the host cell and then break out of the cell and infect other cells. So viruses are categorized based on the way that they use that cellular machinery to replicate their nucleic acids, their genetic material. And the book kind of skims over this a little bit. It doesn't give you all the categories of viruses, so we're going to go in detail a little bit. Obviously, with the COVID-19 pandemic, people are a lot more aware of viruses, a lot more interested in how viruses work. So we're going to go a little more in depth here so you can understand how viruses such as COVID, as well as other types, are able to replicate and reproduce their genetic material. So the first type of virus we'll talk about is known as a DNA virus. Okay. So herpes is an example of a DNA virus. There are a variety of herpes viruses. They can cause cold sores. They can cause genital herpes. So there's a variety of these herpes viruses. But herpes viruses are DNA viruses, which means that they contain DNA, and that DNA is encapsulated by a protein coat. And then once they insert themselves into cells, the coat opens up, releases the DNA, and then that DNA, that viral DNA, is going to undergo two processes. It is going to undergo replication, and that replication is going to make more viral DNA. And of course, that new viral DNA is going to get encapsulated by the proteins that are produced. And so the DNA from our DNA viruses is also going to undergo transcription to produce RNA, right, the messenger RNA. And then the ribosomes inside the host cell are going to translate that messenger RNA into the viral proteins. And then once the viral proteins are produced, those viral proteins can then encapsulate the DNA and make those new viral particles. Okay, so this is how DNA viruses work. Any questions? Okay, the second category of viruses that the book mentions are known as retroviruses. Okay, so perhaps the most commonly known retrovirus, the one that I'm aware of, is HIV. The virus that causes AIDS is a retrovirus. So a retrovirus is going to have RNA inside its protein coat. And the challenge that retroviruses face is that there's no cellular machinery for copying RNA. Right? RNA is formed by the transcription process from DNA, but in a normal cell, your messenger RNA is not copied. So if you have an RNA virus, it has to use some other sort of a process in order to get copies of its RNA so that you can make more viral particles. And so there's two types of RNA viruses, and we'll talk about the retrovirus type first and explain how it is able to get copies of its RNA. Okay, so in a retrovirus, it's going to have an enzyme known as a reverse transcriptase. Okay, so it's going to have a key enzyme called a reverse transcriptase. And that reverse transcriptase, as the name suggests, is going to reverse the process of transcription. It's going to allow DNA to be produced from RNA instead of the other way around. Okay, and so it has this enzyme. So once it's inserted into the cell and the virus particle breaks up, This RNA gets translated to produce this reverse transcriptase enzyme, and that reverse transcriptase enzyme is going to react with some of the remaining RNA and make DNA copies of it. Okay, and then once the DNA is formed, it's going to undergo replication, just like in the DNA viruses, and that's going to make more of the viral RNA. And that viral RNA, once it's formed, two things are going to happen to that. Actually, it's not the replication. Replication may indeed happen, but that's a dead end because the viral DNA isn't used in the process. This should say transcription. Sorry about that. The viral DNA undergoes the transcription process to give viral RNA, and then some of that viral RNA is going to be assembled into the new viral particles, whereas other portions of it are going to undergo translation to give the viral proteins. Okay? So retroviruses get around this problem of not being able to directly copy RNA by using this reverse transcriptase process to convert their viral RNA into DNA. Okay? Any questions on retroviruses and how they're able to reproduce? Yes? It's encoded in the RNA of the virus. So the virus on that mRNA is a gene that encodes for this enzyme. So once it's inside the cell, it uses the ribosome to produce this, and then it's off to the races, and it's able to make the DNA that enables it to make more copies of its RNA. Okay, so this is just part of its RNA. It's encoded for in the RNA. The new viral particles are the ones that enable the virus to infect more cells. A virus has to reproduce in order to continue infecting cells. If it can't reproduce, then the infection just goes away. So this is essential for viruses to be able to copy themselves in order to reproduce. And that's the whole goal of a virus. A virus doesn't have a brain. A virus can't think. But a virus has to, in order to survive, it has to reproduce. And so that's the entire goal of a virus. It's not trying to make its host sick. That's just a byproduct of what it's doing, reproducing. Yes? Yes, so these nucleic acids are not very stable inside of living systems. They're degraded very rapidly, and that's important because once you have the whole cell division process is very tightly regulated, as well as the gene expression process, very, very tightly regulated. So once an mRNA or a DNA has done its job, it needs to be degraded. Okay, in the RNA sense, certainly. And you don't want foreign pieces of DNA floating around in a cell. So the cells are going to destroy that genetic material very quickly, but they need to have this protein capsule. So the viruses have to have that protein capsule in order to protect that genetic material. So mRNA vaccines, which burst onto the scene during the COVID pandemic, use lipid nanoparticles as that shell instead of proteins, because that mRNA inside the COVID vaccines is not very stable. And so the trick, people recognized how useful mRNA would be for vaccines decades ago. But it took at least 20 years to develop all the technology to be able to deliver mRNA to a cell without it getting degraded, to have it survive long enough to get picked up by the ribosome and make the proteins that are necessary for that vaccine to do its job. So the vaccines are using lipid nanoparticles to encode the mRNA, whereas the viruses are using proteins to do that process. But these nucleic acid polymers are not very stable. They get degraded fairly quickly, and that's by design. Okay, good questions. All right, so our third type of virus doesn't really have a good name as far as I can tell. So we'll just refer to them as other RNA viruses. So these are RNA viruses that are not retroviruses, okay? And there are lots of viruses in this category. Colds, flus, COVID, these are all going to be RNA viruses that are not retroviruses, okay? So you might wonder, how does an RNA virus that is not a retrovirus, how does it reproduce itself? So it has to have a special type of enzyme, one that does not exist in host cells, one that does not exist in human cells, and that is an RNA polymerase enzyme. Okay? So RNA polymerase, as I said, is an enzyme that's not part of normal cells, but it allows direct copying of RNA. Okay, so when your RNA, when your viral particle opens up inside of a host cell and releases the RNA that's inside, that RNA is going to get copied by the ribosome. It's going to make a large protein. So typically these RNA viruses, and probably even the DNA viruses, what they do is they make a very long protein chain, and then they have a protease that cuts that protein chain at certain spots to make the individual proteins of the virus. And so one of those proteins is an RNA polymerase that allows copies of the RNA. It allows direct replication of the RNA, and so that's going to make more viral RNA. And then, of course, that RNA is also undergoing the translation process to generate the viral proteins. And then once the viral proteins and the viral RNA are present inside the cell, those proteins can assemble around the viral RNA, making new viral particles that then bust out of the cell and go infect other cells. Okay, so 3 ways that viruses are able to reproduce themselves. In some cases, they can have their own DNA. In other cases, they use this reverse transcriptase process to make DNA inside the cell from their RNA. In other cases, they use this RNA polymerase enzyme to just make direct copies of their RNA inside the cell. Okay? Any questions about viruses and how viruses are able to replicate themselves? Okay, so most antiviral drugs are going to do one of 2 things. They're either going to inhibit viral enzymes specific to the virus. As we mentioned earlier, we talked about nirmatrelvir or Paxlovid, the best COVID drug out there today, which inhibits the viral protease that chops the long polyprotein into the individual proteins. And that's a specific protease to the virus. Or these antiviral drugs will interfere with the replication or the translation process. They'll interfere with the process that the virus uses to either copy its RNA or to produce its DNA. Okay, so that's typically how antiviral drugs will work. Okay, any final questions on viruses? All right, so now we're going to go on to our next chapter, chapter 27. And chapter 27 is entitled Metabolism. And so in this chapter, first we're going to learn what metabolism is, and then we're going to talk about some specific metabolic pathways. So metabolism is a very broad term. Metabolism refers to the sum of all chemical reactions in an organism. Okay? So all of the chemical reactions that are going on inside of you and me would be referred to as metabolism. So we could spend the rest of our lives studying metabolism and we still wouldn't know everything about metabolism. And we're going to spend roughly a day and a half talking about it. So we're just going to kind of scratch the surface of what's going on in metabolism. We can divide metabolism reactions into two processes. So we have catabolism. Catabolism is the breakdown of large biomolecules into smaller ones. Okay. This process typically releases energy, right? Which seems kind of backwards because forming bonds usually is energetically favorable. Here we're breaking bonds and releasing energy. It's not because there's energy stored in the bonds, as we've said repeatedly. It's because of the structures of the compounds that are broken up are designed such that the products are more stable than the reactants. But we're going to talk mostly about catabolism in this chapter, how biomolecules that we digest in our diets, carbohydrates, triacylglycerols, proteins as well, but we're not going to talk about the proteins, are then chopped up into smaller pieces and we get energy from that. But our bodies also do the reverse. Our bodies can also synthesize these larger biomolecules from individual pieces, and that is known as anabolism. Okay, synthesis of larger biomolecules from smaller molecules, and this typically requires energy. Okay. All right, so like I mentioned, we're going to focus primarily on the catabolism process. And we have here, we have a figure, overall figure here from the textbook that describes various stages of this catabolism process, starting from lipids, carbohydrates, and proteins. The process will begin by hydrolysis of those large polymeric biomolecules into the monomeric units. Okay, so with carbohydrates, your polysaccharides such as amylose that you get from starchy materials or other disaccharides if you're eating things with sucrose or lactose and so forth. Those are going to get hydrolyzed by enzymes in your digestive system to monosaccharides. Your proteins are going to be hydrolyzed by protease enzymes that chop the proteins up into individual amino acids. And then your lipids are going to undergo hydrolysis. We'll talk about that in a moment, liberating fatty acids and glycerol. And then in the second stage, each of these monomers is going to get transformed into a common species, namely acetyl coenzyme A, right? So all three of these types of biomolecules that you ingest in your diet are going to ultimately get turned into acetyl coenzyme A. In the case of carbohydrate metabolism and amino acid metabolism, pyruvate is formed first. Pyruvate can be turned into a variety of things, but acetyl coenzyme A is the most common one. And then you'll notice that ATP is produced in the glycolysis pathway. That is, again, an energy storage molecule, as we'll discuss. And then our third stage is going to be the citric acid cycle, where our acetyl coenzyme A is going to be oxidized to give us CO2. CO2 is a very good byproduct because it is a gas. It's very easy for us to excrete CO2. We breathe it out. And then ATP is produced in this process, as well as reduced coenzymes. We'll talk about those in a moment. Those also are able to store energy. And the energy from these reduced coenzymes can be harvested using oxygen in stage 4. So we're not going to talk about all of these things in a day and a half. We're going to focus on three processes. fatty acid oxidation, glycolysis, and the citric acid cycle. We'll focus on those three. We're not going to memorize the entire cycles. That's not our goal here. If you memorize them, you're probably going to forget them in a few weeks. What we're going to do is just help you understand the chemistry that's happening here, understanding the reactions, specifically understanding how reactions we've learned throughout the semester are applied in these processes. So being able to draw reaction mechanisms or conceptually understand reaction mechanisms, being able to predict products from a given intermediate in a cycle, that's what we're looking at. So you do have to have a working familiarity with these cycles, but we're not going to memorize the entire cycles in these processes. So acetyl coenzyme A is a key molecule in all of these cycles. And there's its structure. We're used to just writing coenzyme A, and we might forget that coenzyme A is a massive molecule. You can see it has a nucleotide portion to it. And then it has even kind of a peptide portion to it. But it is coenzyme A is a thiol. So acetyl coenzyme A is a thioester. So what makes acetyl coenzyme A so important? Well, it has two key characteristics. So acetyl coenzyme A is a thioester, and a thioester, of course, is a carboxylic acid derivative. We spent a lot of time in chapter 16 talking about carboxylic acid derivatives. And the alpha hydrogen of a thioester is more acidic than the alpha hydrogen of our other common carboxylic acid derivatives. That would be esters or amides. Okay? So it's going to be easier to deprotonate the alpha carbon of acetyl coenzyme A and make an enolate than it would be for an ester or an amide. And this is going to be important in our metabolic pathways, as we'll see momentarily. And then the second feature of the thioester, acetyl coenzyme A, that makes it useful, remember that all of our carboxylic acid derivatives have a group attached to the carbonyl that can function as a leaving group. And in this case, it would be a thiolate leaving group, a species with a negatively charged sulfur on it. And it turns out that that is a better leaving group than what is present in esters or amides. Okay? So in the reactions we'll see with acetyl coenzyme A, one of these two features is going to be playing a role. The function of that group is a better leaving group, or the ability to make an enolate from acetyl coenzyme A. Okay? And then another key reaction, key process in these metabolic pathways is the hydrolysis of ATP. The hydrolysis of ATP releases energy. It is an exothermic process, and here we see it on the screen. ATP, which we know now is a nucleotide, reacting with water, giving phosphate, inorganic phosphate, plus ADP as a product. Okay, so to me, the easiest way to explain why this process is exothermic is to look at ATP and then look at ADP. Okay, so look at the starting material and one of the two products, right? Inorganic phosphate's the other one. Is there anything about this product, ADP, that looks like it's going to make it more stable or lower in energy than ATP? Yes, Garrett. Okay, so you would say one starting material going to two products. Is that what you're referring to? So there's a hidden reactant, and that is water. So it's actually two starting materials going to two products. So there's something else going on, right? Okay, so you look at the triphosphate group. How many negatively charged oxygens do we have in a triphosphate at physiological pH? We have 4 negatively charged oxygens, and they're fairly close to each other. So that's going to be a destabilizing interaction between those negatively charged oxygens present in ATP. So you hydrolyze it. How many negatively charged oxygens do we have in close proximity in ADP? We have 3. So we've removed one of those negatively charged oxygens. We've reduced the electrostatic repulsion. And that is the main feature that makes ADP lower in energy than ATP. And that's the main feature that makes ATP hydrolysis exothermic. You release one of those phosphates, you are reducing the electrostatic repulsive interactions in that product. Okay, any questions about that? Okay, yes. That's a good question. So you could cleave this one, and that would be more exothermic. My guess is the body needs to shuttle back and forth between ATP and ADP, and it's easier to do that if we've only cleaved one phosphate off. So yes, I would assume that cleaving the second one would also be exothermic, but it would make these metabolic pathways more challenging. It would make it more challenging to go back to ATP. Okay, there are some circumstances where we have AMP. But typically, this is the process that's going on. Okay. All right. So how do we make ATP then? There's a couple different ways that ATP is synthesized. One is the use of creatine phosphate, also known as phosphocreatine. And so this process, and this is how many of these biochemical reactions are written in this coupled reaction form that we saw way back in chapter 6. Creatine phosphate can transfer its phosphate to ADP, making ATP, and then giving creatine as the product of that reaction. And this process is going to be thermodynamically favorable. Why it's thermodynamically favorable, I don't entirely know, to be honest to you, but it is thermodynamically favorable to cleave this phosphorus-nitrogen bond and attach that phosphate onto the ADP. Okay, so that's one way that we can make ATP in nature. Okay. All right, so there are some redox reactions that are performed in metabolism. And we've studied some of the coenzymes that are involved in biological oxidations and reductions previously in the class, namely NAD plus and NADH. This figure in the book that shows oxidation with NAD plus is incorrect. It has a typo. It shows the hydrogen attacking the wrong carbon of this pyridinium ring. So please don't refer to that figure that's in the textbook. As I mentioned, this is these 2 chapters we're doing now, Friday and today and Wednesday, are brand new chapters in the book. They haven't gone through as much proofreading as the other chapters. So there are a few errors in some of the figures, but this is a correct version. This shows NAD plus, and now we recognize NAD plus as also being a nucleotide. So we see nucleotides in a lot of places in metabolism and biochemistry. But it is this nicotinamide or nicotinium ring here that is functioning in the oxidation process. It's serving as an acceptor for H minus that hops over from the alcohol to this coenzyme in that oxidation process. And then there's a base on the enzyme in the enzyme active site that triggers this. And of course, NADH is a byproduct of this. And these biological oxidations can be run in the opposite direction, namely they can be used to reduce aldehydes or ketones. And here we see that. There's also a typo here. These biomolecules are difficult to draw because of so many structures. There's supposed to be an NH2 group here, right? This is supposed to be adenine, and as you know, adenine has an NH2 here. But the business end of NADH is this ring right here, and what happens is it donates its hydride to an aldehyde or a ketone, and then that oxygen gets protonated in the enzyme active site. So this is how oxidation and reduction processes with alcohols, aldehydes, or ketones occur in nature. Any questions about NAD plus and NADH? Yes. Yep. Yep. Yeah, that's a great question. So we talked about it last semester, so, or in 351 or whenever you had 351. The reason H minus functions as a leaving group is it's not really floating around in solution because these molecules are held in very close proximity in the active site of the enzyme. So it's able to just be directly transferred. It's close enough in space that it's able to be directly transferred from this alcohol to this NAD plus molecule. And the driving force there is just neutralizing the positive charge. It's the cost is you're removing aromaticity, but you are also removing that positive charge. So that's why it happens in this fashion. Okay, but because we can't do that in flasks, our oxidation mechanisms that we use in a flask are different from this. Okay, good question. So there's another coenzyme system that performs biological oxidations and reductions. Before we do that, we'll just look here. This is again how a common oxidation reaction would be drawn in a metabolic pathway. The coenzyme, the products from the coenzyme, the name of the enzyme that's used. Dehydrogenase is biochemistry term for oxidation because you're removing hydrogen. That's one of the ways that we can oxidize a molecule. And here we're oxidizing the alcohol of malate into a ketone of oxaloacetate. Our second coenzyme system that performs oxidations in nature is FAD. That's the oxidant, and FADH2 is the reducing agent. And you can see FADH2 is the reduced form of FAD. Why? How do we tell it's reduced? It has more hydrogens and fewer pi bonds. That's how we can tell this is the reduced form. This is the oxidized form with fewer hydrogens and more pi bonds. Okay, so how do FAD and FADH2 work? Well, we're going to focus on FAD because we're going to see it performing an oxidation in the fatty acid beta oxidation pathway that we're going to talk about momentarily. So what happens is a fatty acyl coenzyme A derivative is going to first be deprotonated by a base inside the active site of the enzyme. Remember, I told you this is easier than it would be with esters or amides. That alpha hydrogen of a thioester is more acidic than the alpha hydrogen of an ester or an amide. So in the active site of an enzyme, we're able to make an enolate of a thioester. And then that enolate of a thioester is going to eliminate out hydride. Have we seen enolates eliminate leaving groups from beta carbons before? Okay, we saw it in the dehydration of the aldol reaction, where an OH group was eliminated in an E1CB mechanism. So this is an E1CB process, but instead of eliminating an OH, what we're eliminating is H minus, which of course we can't really do in a flask, but because this enolate is held in very close proximity to the FAD molecule in the active site of the enzyme, this H minus hops onto the nitrogen, the electrons are pushed in this fashion, and then there's an acid in the active site that donates a proton to the other nitrogen, okay? So that's going to give us our alpha beta unsaturated fatty acyl coenzyme A derivative, alpha beta unsaturated thioester, and then the reduced form, the FADH2. So in general, in nature, NAD plus is going to oxidize alcohols, whereas FAD is going to oxidize other functional groups. Okay, so usually we'll see NAD plus for alcohol oxidations, FAD for oxidations of other functional groups, such as in this case, oxidizing a carbonyl compound to an alpha beta unsaturated carbonyl compound. Okay, any questions about FAD and FADH2? So the reduced forms, in addition to being able to function as reducing agents, they are also sources of energy. They can use the electron transport chain. They're electron-rich species, and they can donate their electrons to generate energy. And a single NADH molecule is going to produce 2.5 equivalents of ATP. Okay, you might wonder how it can get half an equivalent of ATP. We're not going to go into that. We're just going to understand the energetic equivalence here between these reduced coenzymes and ATP molecules. And then FADH is going to be able to deliver energy equivalent to 1.5 equivalents of ATP. So these molecules are sources of energy as well. All right, so now we're going to talk about the beta oxidation process, or how triacylglycerols are catabolized in nature with the release of energy. And there are 3 parts to the process. Okay, part 1 is an ester hydrolysis. Triacylglycerols have esters, they're triesters, and so the first step in their catabolism is going to be the hydrolysis of these triacylglycerols. Okay, so the triacylglycerols going to look like this. These groups may or may not be the same. We'll just go ahead and draw them all as being R. And so the ester hydrolysis is going to involve water. It's also going to involve an enzyme known as a lipase. It's going to catalyze that hydrolysis. And what we're going to end up with is glycerol. Glycerol is this trialcohol, this 3-carbon trialcohol. And then we're going to end up with 3 molecules of a carboxylic acid. Okay, so that's part 1 of the beta oxidation pathway. Okay, and then part 2 is going to involve the glycerol, what the glycerol gets turned into, catabolism of glycerol, and that's a 2-step process. We're going to use a kinase. Kinases are enzymes that attach phosphate groups. So the kinase is going to attach a phosphate group to the glycerol, giving glycerol 3-phosphate. And now we're going to have one of these oxidations we talked about. This is going to involve NAD+, because it's oxidation of an alcohol. So NAD+ is going to use that mechanism we showed you earlier, deprotonating that oxygen, hydride being transferred to NAD+, giving you NADH, an energetic molecule, as well as dihydroxyacetone phosphate. This is important because, as we will learn on Wednesday, dihydroxyacetone phosphate is part of the glycolysis pathway. So this can then be metabolized or catabolized more using glycolysis. But then part 3 of the process is going to involve the beta oxidation. So it's going to explain the fate of the carboxylic acids in this process here. So we're going to have a long chain fatty acid. I'm not going to draw the whole chain. We're going to use R groups. And first we have to convert that long chain fatty acid into a thioester. A thioester of coenzyme A, okay? And then that thioester of coenzyme A is going to be chopped up to give acetyl coenzyme A. So we're going to chop off 2 carbons, acetyl coenzyme A, and then we're going to get a fatty acid thioester with 2 fewer carbons. Okay, we've chopped out 2 carbons from this to give that. And then that process, part 3, goes over and over and over again until we have turned our long chain fatty acid into a bunch of acetyl coenzyme A's. Okay, so we've said that fatty acids are even in number of carbons. This is the reason they're even in number of carbons, so we can produce these coenzyme A, these acetyl coenzyme A species. So how is the thioester made? The thioester is made by using an acyl phosphate. So this carboxylic acid gets turned into an acyl phosphate. And it's going to be, we'll just write adenosine here. It's an adenosine monophosphate. So this carboxylic acid reacts with ATP, knocking off the diphosphate of ATP and giving an adenosine monophosphate. And then that reacts with coenzyme A to make that thioester. All right, well, it's time to stop here. We will finish the fatty acid beta oxidation and then do glycolysis and the citric acid cycle on Wednesday.